This information is brought to you by Charles Sturt University.
Alright, Paul, can you hear and see everything okay? You're not coming up on my computer screen that it's telling me it's not connecting, but I can hear you there. Paul, can you guys hear me and see the screen?
Yeah.
Okay, cool. Alright, so I'll pick up from where we started this morning. So finishing off with tubular reabsorption. Sorry about cutting off soon, I thought I went through to the full hour. Tubular reabsorption mainly consists of sodium reabsorption, which is the key to everything else. So what we were finishing up talking about there was the two different types of transport proteins that are responsible for sodium uptake. So these ones are the symports and the antiports. So symports bind sodium with another solute such as glucose, amino acids or lactate, while antiports are the sodium and hydrogen transport system. When sodium comes into a cell, hydrogen will come out. So it kind of works over each other there. So we've got sodium coming in and then hydrogen coming out. So sodium is prevented from accumulating in the epithelial cells by the sodium-potassium pumps on the basal surface of the epithelium. So it pumps sodium out into the extracellular fluid, which is then picked up by peritubular capillaries and returned to the bloodstream. So remember where those peritubular capillaries sit is intertwined around the convoluted tubules. So because they're in that location and they're nice and close to the convoluted tubules, they're then able to pick up on the sodium that has been pumped out of the cell by that sodium-potassium pump. So potassium, magnesium and phosphate ions diffuse through the paracellular route via water. So this is with solvent drag. Phosphate is also co-transported into the epithelial cells with sodium, and some calcium is reabsorbed through the paracellular route in the proximal convoluted tubule, but most calcium occurs later in the nephron. So this here is the reabsorption in the proximal convoluted tubule, where water and solutes in the tubule are carried through the epithelium by the symport method, so binding to sodium, or by the antiport method, so when sodium comes in, hydrogen will come out. So also there are acroporons. So just here, acroporon is the escaping of water between the channels, and the paracellular route is between the cells. So from here, as they've got sodium, glucose, and hydrogen, and also anions as well, so positively charged ions coming, or most of them are coming out of the tubular fluid and then back into the bloodstream. So this exchange has taken place either through the cell, which is the cellular route, or then through the gaps in the middle, so in between the tight junction there between the two different epithelial cells there, via the transcellular route. So because we've got the exchange of all of these necessary ions, necessary, I guess, for human functioning, they can then get put back into the peritubular capillary that's waiting around the outside of the convoluted tubules, and then back into the bloodstream to be distributed around to the rest of the body. Glucose is co-transported with sodium by sodium glucose transport proteins, or SGLT proteins. So urea, which is a nitrogenous waste, diffuses through the tubule epithelium with water, and then reabsorbs 40 to 60% in the tubular fluid. So because we've got so much water coming in to transport all of those other ions back into the tubular fluid, we've then got a whole lot of water that's still in the tubular fluid, which would mean if all of this went through all the way through to the urine phase, we'd be excreting an enormous amount of urine per day. So there's still a lot of this water reabsorption to take place, where the water will then transport these other ions here, and then getting them outside the cell back into peritubular capillary, where it will then have its influence on blood volume and other factors associated with the what's going through the blood flow. So kidneys remove about half the urea from the blood, and creatinine is not reabsorbed at all. Instead, when creatine gets into the tubular fluid, it'll then stay there and then be excreted via the urine. So kidneys reduce 180 litres of glomerular filtrate to about 1 to 2 litres of urine per day. 2/3 of water in the filtrate is reabsorbed by the PCT, proximal convoluted tubule, and then reabsorption of all salt and organic solutes make the tubule cells and tissue fluids more hypertonic. So hypertonic means there is more solutes in the fluids of the proximal convoluted tubule, and this is causing a higher osmotic pressure. So after water and solutes leave the basal surface of the tubular epithelium, they are reabsorbed by the peritubular capillaries. So they, essentially they escape through the gaps of the tubular cell wall, so where the gaps were in that in those epithelial cells. This is where it'll escape through those walls, and then reabsorbed back into the bloodstream by osmosis and solvent drag. So there are 3 factors that promote osmosis into the capillaries. So the first one is interstitial fluid pressure. So there's high interstitial fluid pressure that drives water into the capillaries. So this is caused by a build-up of all the other reabsorbed fluids. Okay, so the more reabsorption of the fluids that there are, the more fluids that are available in that tissue space, and then the build-up of pressure. Then we've got the narrowness of the efferent arteriole. So remembering the bottleneck effect in that we've got greater build-up behind the efferent arterioles because we've got greater blood flow in and then less of a blood flow out, which lowers blood hydrostatic pressure and causes less resistance to absorption. And then we've got proteins which are made in the blood after filtration, which elevates the COP, the colloid osmotic pressure, so the pressure that's associated with protein dragging and creating pressure. So high COP and low BHP in the capillaries, when it leaves the glomerulus, and a high hydrostatic pressure in the tissue fluid, the balance of the forces in the peritubular capillaries favours absorption. So think of the peritubular capillaries at the top of the priority list when it comes to the COP and the BHP, so the colloid osmotic pressure and then blood hydrostatic pressure too, and then the peritubular capillaries essentially are favouring that absorption. The protocol for the fluid is, or for the pressure, is to get the osmotic pressures and promote osmosis of water back into the bloodstream. Next is tubular secretion. So this is the process in which renal tubules extracts chemicals from capillaries and secretes them into the tubular fluid. So there are two purposes in the proximal convoluted tubule and the nephron loop. These are removing waste and then also having the acid-base balance that obviously we'll go over more so this afternoon when we get to that lecture. So in waste removal, this includes urea, uric acid, bile acids, ammonia, catecholamines, prostaglandins, and a little creatinine are secreted into the tubule. Secretion of uric acid compensates for reabsorption earlier in the proximal convoluted tubule. Okay, so because uric acid is picked up in the proximal convoluted tubule, so that first port of call after it comes out of the glomerulus and has been filtered, we then have to compensate for that action and then get pull it back into the urine stream or the bloodstream, back into the urine stream later on through that nephron. So when you get to the nephron loop and then also the distal convoluted tubule, that's when we start to pull in more of those wastes that have been reabsorbed back into the bloodstream. Also clears the blood of pollutants, morphine, penicillin, aspirin and other drugs. So the way that these are cleared out needs to be kind of somewhat regulated in that when we've got the presence of these drugs in our system, they won't all be excreted in one go. Instead, the body has its somewhat of a homeostatic effect in terms of trying to bring it back to a nice, even or steady balance back down to a resting level. And then the other purpose is the acid-base balance, which is the secretion of hydrogen and bicarbonate ions to help regulate the pH of body fluids. So maintaining a normal pH balance, so not too acidic and not too basic, and then that will help with the excretion of other solutes in through the urine. So the primary function of the nephron loop is to generate salinity gradient that enables the collecting duct to concentrate the urine and conserve the water. So by concentrating the urine, what that means is basically getting rid of any excess water out of that collecting duct and then maintaining or before it reaches the collecting duct, and then maintaining all of those solutes and waste and uric acid, so keeping that and increasing the concentration of the urine, keeping that inside the nephron, and then getting rid of the water and putting that back into the bloodstream where it can then regulate blood volume by either increasing or reducing the blood volume that's necessary depending on how hydrated or dehydrated you are. So electrolyte reabsorption from filtrate. So the thick segment of the nephron loop reabsorbs 25% of the sodium, potassium and chloride, and these ions then leave the cell by active transport and diffusion. So sodium chloride remains in the tissue fluid of the renal medulla once it's been excreted from the thick segment of the nephron loop, where water cannot follow because the thick segment is impermeable to water. So when we talk about the sodium chloride remaining in the renal medulla, so this is the area where we've got the renal pyramids and the renal columns. And then other than that, we've got water which is impermeable to the cells of the thick segments of the nephron loop. So tubular fluid is very dilute as it enters the distal convoluted tubule. So the further along the process it goes, so the more reabsorption that takes place and the more secretion that takes place as well along those stages of the nephron, then account for the increasing concentration of the urine and then the reduction in water content too. So we've got a higher amount of solutes for a lower amount of volume. So from the distal convoluted tubule and collecting duct, so fluid arrives in the distal convoluted tubule still containing about 20% of water and 7% of salts from the glomerular filtrate. So remember how I said just before that we still have a lot of water reabsorption to take place. So we need to get the water out of the nephron and back into the blood system so that then we can have a maintaining or a balance of our blood volume. So if all of this was passed through, we would amount to having about 36 litres of urine per day. So obviously there is still a lot of reabsorption to take place. So the distal convoluted tubule and collecting duct reabsorb variable amounts of water, salt, and then are regulated by several hormones. So these include aldosterone, atrial natriuretic peptide, and then antidiuretic hormone, and then parathyroid hormone there too. So aldosterone is the salt-retaining hormone. This is a steroid secreted by the adrenal cortex. So the adrenal cortex are the glands that sit on top of the kidneys. So when blood sodium concentration falls, or when potassium concentrations rise, this is when aldosterone will be released from those adrenal cortexes. So by having a reduction in the blood pressure, aldosterone is then released, and then what aldosterone does is it has an impact on the angiotensin 1 turning into angiotensin 2, which then tells the body to retain as much fluid as possible. And then that in turn will improve blood volume and then improve blood pressure. So remember when we've got the sodium and then potassium pumps too. So as the sodium comes in, potassium comes out, so it'll be able to regulate the amount of salt that's either being reabsorbed or excreted through the urine. So aldosterone acts on the thick segment of the nephron loop, the distal convoluted tubule, and then the cortical portion of the collecting duct. So the portion of the collecting duct that's still sitting up the top in the renal cortex before it comes into the renal medulla. So this works to stimulate the reabsorption of more sodium and then the secretion of potassium. Okay, so aldosterone's main aim essentially is to regulate the amount of salt that we have floating around in the body. And then we've got AMP. So AMP is secreted by the atrial myocardium in response to high blood pressure. So myocardium being the muscle of the heart. So there's 4 actions of ANP that result in the excretion of more salt and water in the urine. So this will then in turn reduce blood volume and pressure. So it dilates the afferent arteriole and constricts the efferent arteriole. So this in turn will, because we've got the afferent arteriole being able to deliver more fluids, and the efferent arteriole is going to be then less able to take more away. Okay, so we've got more arriving than what we're getting taken away. We've got an increase in the glomerular filtration rate. And this is because we've got more fluid arriving than what's being taken away. Inhibits renin and aldosterone secretion. So renin is what comes from the kidneys that turns aldosterone or helps the angiotensin 1 to turn into angiotensin 2. Inhibits the secretion of the antidiuretic hormone and inhibits sodium reabsorption by the collecting duct too. So we've got the AMP, which works in response to high blood pressure. So by doing this, we want to get more fluid out of the body. So it's increasing the rate of urine formation, so we can get it out of the body and then bring blood pressure back down to a normal resting rate. Antidiuretic hormone is secreted by the posterior lobe of the pituitary, and this is released in response to dehydration and raising osmolarity, which stimulates, so raising osmolarity then stimulates the hypothalamus. The hypothalamus then acts on the posterior pituitary to then release the antidiuretic hormone. So this makes the collecting duct more permeable to water, where water in the tubular fluid re-enters the tissue and the bloodstream rather than being lost in the urine. And then we've got the parathyroid hormone. So the parathyroid hormone basically works to restore the homeostasis of calcium in the body. So this is secreted from the parathyroid glands in response to calcium deficiency or hypocalcemia. Acts on the proximal convoluted tubule to increase phosphate excretion, and then also acts on the thick segment of the ascending limb of the nephron loop, so the limb that's coming back up, the thick segment coming back up right before it gets into that distal convoluted tubule. Also acts on the distal convoluted tubule and increases calcium reabsorption. So this helps to minimise a further decline in the calcium levels. We also got an increase in phosphate content, which lowers the calcium content in urine. So essentially we're excreting less calcium, therefore we've got more in the body, so that's how it's going to help regulate the amount of calcium declining. And then the parathyroid hormone also stimulates calcitrol synthesis by the epithelial cells of the proximal convoluted tubule. So the proximal convoluted tubule absorbs about 65% of the glomerular filtrate, so the stuff that's been filtered through the glomerulus, and then returns it to the bloodstream via the peritubular capillaries, depending on what solutes we've got in the blood. So things like hydrogen will get excreted, while sodium, depending on salt levels in the blood, will either get excreted or reabsorbed back into the bloodstream in through those peritubular capillaries. All right. So now move on to the next lecture, so water and electrolyte balance. So a newborn baby's body weight is about 75% of water. So as the baby starts to excrete and get rid of some of this excess water, its weight can start to reduce after the first day or so. Young men have an average of 55 to 60% of their body mass is water, and then total body water, or TBW, of a 70 kilogram young male is around 40 litres. So all of the body's water, so that 55 to 60% of the body water we had, is all separated into different fluid compartments. So these are all separated by permeable membranes. So we've got 65% of that water is intracellular fluid, 35% of it is extracellular fluid, which is then further segmented down into tissue fluid, blood plasma and lymphatic fluid, and transcellular fluid, so the fluid that's going in the cells. So of that transcellular fluid, this then accounts for cerebrospinal fluid, synovial fluid, peritoneal fluid, pleural and pericardial fluids. Also is in the humors of the eye, so it's just basically a fluid compartment of the eyeball. And then also accounts for bile and fluids of the digestive, urinary and reproductive tracts. So still talking about the fluid compartments. So fluid is continually exchanged between compartments. This is done via through capillary walls and through plasma membranes as well. So from tissue fluid, water may be reabsorbed via capillaries, and then from capillaries into cells via osmosis or into the lymphatic system. So all of those different compartments are exchanging the amount of fluid that they have. Because water moves so easily through plasma membranes, osmotic gradients never last for very long. So between the intracellular fluid and the extracellular fluid, any imbalances can be restored very quickly. So this is done through those homeostatic pathways. So as soon as the body detects that there's either an increase or a reduction in water, it then activates mechanisms that oppose it to then get it to a nice even balance. So if imbalances arise, osmosis restores balance within seconds, so the intracellular and extracellular fluid are equal or at that homeostatic balance. If osmolarity of tissue fluid rises, water moves out of the cell, and then vice versa. If osmolarity of tissue fluid falls, water will then move into the cell. Osmosis. So osmosis from one fluid compartment to another is determined by the relative concentration of solutes in each compartment. So electrolytes are the most abundant solute particles, and those are comprised of sodium salts in the extracellular fluid and then potassium salts in the intracellular fluid. Electrolytes play a principal role in governing the body's water distribution and total water content. Okay, so you can start to see how water balances and intracellular and extracellular fluid kind of balance out nicely with what we've just spoken about with the urinary system in terms of reabsorption of those salts back into the bloodstream. So fluid balance. So daily losses, so gains and losses are supposed to be somewhat equal with about 2.5 litres per day. Gains can come from two sources, so preformed water or metabolic water. So preformed water is just essentially digested water, so this can come from foods or drink. And metabolic water is where H2O or water is a byproduct of aerobic metabolism and dehydration synthesis. Okay, so that accounts there for 200 mils of the total 2.5 litres of intake per day, with food and drink equating to about 2.3 litres per day. And then we'll go through the output later. So the regulation of fluid intake, so mainly governed by thirst, which is controlled by the following factors. So we've got dehydration and then also the osmoreceptors in the hypothalamus. So dehydration reduces blood volume and blood pressure. This is because we've got a reduction in readily available fluids in the body, and also increases blood osmolarity. So because we've got a reduction in volume and then an increase in the number of solutes, this causes an increase in the osmolarity. So osmoreceptors in the hypothalamus. Osmoreceptors are sensory receptors which receives information about osmolarity in the body. Okay, so if your osmoreceptors are tingling, essentially means that you've got high osmolarity throughout the body. So osmoreceptors respond to angiotensin II, which is produced when blood pressure drops, and to rise osmolarity in the extracellular fluid with a drop in blood volume. So remember, angiotensin II works to vasoconstrict blood vessels, and then also works to have an impact on the kidneys and the nephrons to increase fluid reabsorption or water reabsorption from the nephron. Osmoreceptors communicate with the hypothalamus and cerebral cortex, where the hypothalamus produces antidiuretic hormone and just tells the body to retain water. So there's signals coming from the brain to the kidneys that say don't lose any more water through urine, we need to hang on to as much of that as possible because we're starting to feel a bit thirsty. And then we've got the cerebral cortex, which produces the conscious sensation of thirst. So with intense thirst and a 2 to 3% increase in plasma osmolarity, or 10 to 15% of blood loss. So a 2 to 3% increase in plasma osmolarity is essentially a significant reduction in blood volume, which is then going to start to send signals to those osmoreceptors and tell the brain to send out signals to the rest of the body to distribute that conscious sensation of thirst. So you feel like drinking water and you have a drink, and then also will tell the body to lose salt and retain water. So this here is just a picture kind of indicating what happens when we're dehydrated. So when you're dehydrated, you'll have an increase in blood osmolarity, and then also a reduction in blood pressure. So this goes hand in hand in that blood osmolarity goes up when we've got a reduction in volume, increase in solutes. And then because we've got that reduction in volume, it then in turn reduces blood pressure. So in the kidneys, when the kidneys detect that we've got that reduction in blood pressure, the kidneys release renin, which then acts on the angiotensin I, which then transforms into angiotensin II, which stimulates the osmoreceptors. Okay, so when we've got angiotensin II in the body, that's essentially the body telling itself that it's thirsty and that we need to retain as much water as possible. So when the osmoreceptors stimulate the hypothalamus, we then get a reduction in the amount of saliva, which is caused by the inhibition of salivary glands, and then also lower capillary pressure. So because we've got lower blood pressure, we've then got lower capillary pressure, meaning we're less capable of producing as much saliva as what we normally would. The production of saliva as well, something interesting to note, varies among individuals. So some people can and some people won't produce as much as the other. So when we get a reduction in salivation, this then causes us to have a dry mouth and then leads to that sensation of thirst. So the sensation of thirst will then lead us, hopefully, to go and have an ingestion of water, which then cools and moistens the mouth, rehydrates the blood, so increasing that blood volume, and then distends stomach and intestine. So short-term inhibition of thirst and then the long-term inhibition of thirst. So essentially what these ones are telling you is that how long your body is able, or how long it takes your body to be able to ingest the water and then get it out to the rest of the body, or distribute it around back to the blood volume as quickly as it possibly can, so to stop the signals from those osmoreceptors telling the brain that we're thirsty and we need to retain as much water as possible. That depends there on how long your body is, or how efficient your body is to get rid of the water and distribute it around the body as it's necessary. So now talking about the output of water. So the only way to control water output significantly is through the variation of urine volume. So when we're saying that kidneys cannot replace water or electrolytes, essentially what this means is the kidneys don't generate their own amount of water, or they don't generate their own electrolytes. What they do is just basically distribute the amount that's either going into the body, staying in the body, or then being excreted with urine. So because we can't generate our own water that's inside the body, we then have to replace that with having a drink of water. So the mechanisms involved in the regulation of the urine output essentially are changes in urine volume, which are linked to adjustments in sodium reabsorption. So as sodium is reabsorbed or excreted, water will then follow. So that works kind of on a proportionate pathway. So then with the amount of sodium that's being excreted or reabsorbed, water kind of goes hand in hand with that relationship there too. So the concentration of urine is done through the action of the antidiuretic hormone, where antidiuretic hormone controls water output outside of the sodium changes. So essentially they work independently of each other. So the antidiuretic hormone secretion is stimulated by the hypothalamic osmoreceptors, so the sensors in your brain that tell you that your blood volume is low, blood pressure is dropping, and this happens in response to dehydration. So aquaporins, so what the aquaporins are when there's a substance that's dissolved in the water to get it through a cell membrane. This is then synthesized in response to antidiuretic hormone. So the aquaporins kind of act as like gatekeepers of water to get it in and out of the cell. So they're like the gates that sit on the outside that either say yes or no to the water getting into that intracellular fluid. So this here is just showing you how the regulation of the antidiuretic hormone works. So from the pituitary pathways, and shown in response with these red ones here, are showing you the negative feedback loop. So when we've got a reduction in water intake and then an increase in sodium in the body, this elevates blood osmolality, so a reduction in water volume and then an increase in the solutes, which stimulates the hypothalamic osmoreceptors and then stimulates the posterior pituitary to release the antidiuretic hormone. So then where we get that feedback loop kicking in is that when the body tells itself that we're low on water, we then start to feel thirsty, we'll then have a drink, and then that's a negative feedback loop kicking in because we've got an increase in water there. So essentially we've voluntarily kicked in our own negative feedback loop. So we've started to generate mechanisms that oppose that thirst feeling by having some water. So from the stimulation of the posterior pituitary and the release of the ADH, which stimulates the distal convoluted tubule and collecting duct to increase water reabsorption. So if we're unable to get to water to have a drink as soon as possible, the body then tells the DCT and the collecting duct to increase the amount of water reabsorption, so reduce the amount of water that we're losing through urine. Okay, electrolyte balance. So the physiological function of electrolytes is that they're chemically reactive and participate in metabolism. So this then determines the electrical potential or different charge across cell membranes. And then therefore will have a strong effect on the osmolarity of body fluids and affect the body's water content and distribution. So major cations are positively charged ions. So these are sodium, potassium, calcium and hydrogen. You can see them all there. They're all positively charged. And then the major anions, so negatively charged ions, are chloride, bicarbonate and phosphate. So sodium. Sodium is So sodium the principle responsible for resting membrane potential. So remember where we've got the sodium-potassium pump, so sodium comes in, potassium comes out. So this is the principal cation in extracellular fluid, so they account for 90 to 95% of osmolality of the extracellular fluid. So most significant solute in determining total body water and distribution of water among the fluid compartments. The sodium gradient is a source of potential energy for co-transport of other solutes such as glucose, potassium and calcium. So this equation here, or this ion, is sodium combining with bicarbonate, which has a major role in buffering the pH in the extracellular fluid. So because sodium controls the acidity or the base of the pH in the extracellular fluid, by combining with bicarbonate, it's then able to essentially get rid of the hydrogen ions and then keep a hold of the sodium that may be needed or necessary around the body. So an adult needs about 0.5 grams of sodium per day. The average American male diet, I think they consume 3 to 7 grams per day. So to have a sodium deficiency is very rare. And then the sodium concentration is coordinated by aldosterone, which is labelled as the salt retaining hormone. So aldosterone receptors in the ascending limb of the nephron loop, the distal convoluted tubule, and the cortical part of the collecting duct. So essentially the sodium plays a major role in the adjustment of sodium. So if we're keeping it and excreting it, or if we're reabsorbing it and putting it back into the bloodstream. So antidiuretic hormone modifies water excretion independent of sodium excretion. So where we've got this AMP and BNP, they inhibit sodium and water reabsorption, and then the secretion of renin and antidiuretic hormone. And potassium, so this is the most abundant intracellular fluid. It produces with sodium the resting membrane potential of action potentials across the plasma membrane, and are the greatest determinant of intracellular osmolality and cell volume. So when we're talking about the sodium-potassium pump, obviously we need sodium and potassium to be able to actually carry out the functions of that sodium-potassium pump. So potassium is a cofactor for protein synthesis and other metabolic processes, and then also functions with the thermogenesis, so the production of heat as well. So potassium homeostasis is closely linked to that of sodium in that along the proximal and distal convoluted tubule through the nephron, and then the collecting duct, whether it's excreted with urine or then reabsorbed back into the bloodstream. And 90% of potassium in the glomerular filtrate is reabsorbed by the proximal convoluted tubule. Okay, so when it passes from the glomerulus, then goes straight through into the PCT. And chloride is required for the formation of stomach acid. So this is hydrochloric acid, which is the most abundant anion in the ECF, so extracellular fluid, and major contributor to ECF osmolarity. So the chloride shift that accompanies carbon dioxide loading and unloading in red blood cells. So obviously the chloride is going to have a major impact on the chloride shift, which is a form of maintaining the blood pH. And then primary homeostasis is achieved as an effect of sodium homeostasis. So as sodium is retained, chloride ions will passively follow. Okay, so as soon as the body tells itself to start retaining some sodium from the kidneys, the chloride ions will then follow in the same fashion as well. And then calcium. So calcium lends strength to the skeleton, obviously, you should know that by now, and activates the sliding filament theory and the mechanism of muscle contraction. So where we've got the myosin and active playing with each other, and then the binding that takes place when we've got calcium present in the sarcoplasmic reticulum. This serves as a second messenger for some hormones and neurotransmitters, and activates exocytosis of the neurotransmitters and other cellular secretions. So this is the act of moving proteins into and out of a cell. Calcium is also an essential factor in blood clotting. Homeostasis is chiefly regulated by the parathyroid hormone, calcitrol and calcitonin when it's in children. And these hormones then affect bone deposition and reabsorption, and then intestinal absorption and urinary excretion. So phosphates, so they're relatively concentrated in the intracellular fluid due to hydrolysis of ATP and other phosphate compounds. So every process that relies on ATP in the body will then rely on these phosphate compounds. So these activate many metabolic pathways by phosphorylating enzymes and substrates such as glucose. So it's just the breaking down of those substrates is done by the phosphorylation. Phosphates also work as buffers that help to stabilise the pH of body fluids. So a normal diet, given that we consume well over the amount of sodium that we're supposed to, given the the statistic about the American male, the normal diet will provide enough phosphates as well to be picked up by the small intestine to then assist with the regulation and the stabilisation of the pH. So normally phosphate is continually lost by glomerular filtration, but because we're constantly consuming enough phosphates, we won't need to worry about nutritionally supplementing that loss. And then if plasma concentration drops, the renal tubules reabsorb all of the filtered phosphate. So for whatever reason, if we have a lack of phosphate, it is then able to be reabsorbed back into the bloodstream from the nephron. So blood pH, it's solely determined by the amount of hydrogen ions that we have in the body, and one of the most important aspects of homeostasis. So any slight deviation from the normal blood pH of 7.35 to 7.45 can then cause, catastrophic shutdown or interference with other metabolic pathways too. So metabolism depends on enzymes that are sensitive to blood pH. Slight deviation from the normal blood pH can shut down entire metabolic pathways. And slight deviation from normal pH can also alter the structure and function of many macromolecules. So changes to acid-base balance that might occur, so if we ever deviate from the 7.35, 7.45, is because during metabolic pathways, we're then producing a whole lot of lactate or lactic acid essentially from following all these different pathways. So we're generating lactic acid from anaerobic fermentation, phosphoric acid from nucleic acid catabolism, and then fatty acid breakdown as well, so catabolism there, and then carbonic acid is generated from carbon dioxide. So with those examples there, we're then producing a whole lot of additional hydrogen ions, which is then going to impact on the blood pH. So the body's got to be really effective at balancing out the amount of hydrogen that we have in our system to maintain that 7.35 to 7.45 on the normal pH range. So acids. Acids are any chemical that releases hydrogen in a solution. So strong acids, so we've got strong acids and weak acids. So strong acids, such as hydraulic acid, ionize freely, which means it gives up most of its hydrogen ions. So when it ionizes freely, it's happy to give up its hydrogen ions. Doesn't need it, that's okay, it can pass it along, which then markedly lowers the pH of a solution. So then we've got the weak acids, such as carbonic acid, which ionize only slightly. So it's chemically bound to its hydrogen ions, so it's not so willing to let go of those hydrogen ions. But because of this, it will markedly make any difference to the blood pH. Okay, so we've got the strong acids and the weak acids. Bases are any chemical that accepts hydrogen. So when we've got the strong acids that release the hydrogen ions, so get rid of it, doesn't need it, happy to pass it along, the bases will then pick that up. So there are strong bases and there are weak bases. So strong bases, such as a hydroxide ion, have a strong tendency to bind hydrogen, which then markedly raises pH. So you can see homeostasis and trying to keep that stable balance. So where we've got the strong acids losing hydrogen, we've got the strong bases that then pick it up. And then we've got the weak bases, such as the bicarbonate ions, so this one here, HCO3, which bind less available hydrogen and then have a less effect on pH. So essentially with the weak bases and the weak acids, they don't have much of an influence on blood pH, basically because they don't really want to give up or take on any extra hydrogens. And then with the strong bases and then the strong acids, they're willing to let go and pick up any additional hydrogen ions. So the buffers are any mechanism that resists changes in pH. So this converts strong acids or bases to weak ones. Okay, so where we've got strong ones, meaning maybe we've got a higher level, so if we've got a strong base, we've got lots of hydrogen ions that are ready to be released and essentially will be excreted out with urine. So there's physiological and chemical buffers. So physiological buffers are systems that control the output of acids, bases, or carbon dioxide. So this includes the urinary system and the respiratory system. So the urinary system buffers the greatest amount of acid-base balance, so essentially it can happen more of it can get buffered through the urinary system, however it can take a lot longer. So it can take a couple of hours or up to a couple of days, where in the respiratory system it'll buffer within minutes, however it's not as much of a buffering process takes place, so it's not able to actually coordinate all the hydrogen ions as quickly as the urinary system, as often as the urinary system does. And then we've got chemical buffers, which are a substance that binds hydrogen and removes it from the solution as its concentration begins to rise, or releases hydrogen into a solution as its concentration begins to fall. So this restores normal pH in fractions of a second, and there are three major chemical buffers. We've got bicarbonate, phosphate and protein systems. So these mixtures are all referred to as buffering systems. Okay, so they're able to take on hydrogen and then release it into a solution where it might be necessary. So it can take hydrogen that's freely floating around and then put it somewhere where it might be necessary or needed for human function. So the renal control of pH, so the kidneys can neutralise more acid or base than either the respiratory system or chemical buffers, but again this happens at a slower rate. So in the renal control in the kidneys, it can take a couple of hours or up to a couple of days. So the renal tubules secrete hydrogen into the tubular fluid, which bind bicarbonate, ammonia and phosphate buffers, so those buffering systems, where bound and free hydrogens are excreted in the urine, are actually expelled from hydrogen from the body. So we're reducing the amount of hydrogen ions we have in the body by expelling it in urine, which occurs a lot more than what it would in the respiratory system or via the chemical buffering. Other buffer systems only reduce its concentration by binding it to other chemicals. Okay, so essentially instead of getting rid of it, it's just passing it on. So if you don't want to do one of your jobs at home and you're kind of, can't think of the word, passing it on to someone else to do it instead of you having to do it yourself. So that's what the other buffering systems do, is they're just basically transporting hydrogen ions around as opposed to actually excreting it and getting rid of it in the body via urine. Alright, final slide is looking at this one here is the secretion and neutralization of hydrogen ions in the kidney. So hydrogen in the blood reacts with this here, HCO3, which is the bicarbonate, to form H2CO3, which is carbonic acid. So carbonic acid decomposes into water and carbon dioxide, which then enters the tubule cell. So the tubule cell here is the one, the stream of epithelial cells that's running on the outside of the proximal and distal convoluted tubules. Tubule cells acquire carbon dioxide from blood, tubular fluid and their own aerobic respiration. So remember we've got the metabolic production of water that takes place. And then we've got carbonic anhydrase, so CAH, combines water and carbon dioxide to reform this H2CO3, so this carbonic acid formation, the carbonic acid molecule. So the carbonic acid then ionizes to form bicarbonate, which returns into the blood and then separates its hydrogen ions. So I don't know if you can see it very well there. This is just straight out of your textbook as well, so you can have a flick back if you'd like to. Having a look at where the hydrogen ions are and where they're kind of free-flowing. So the antiport, which is taking sodium and then losing hydrogen, exchanges hydrogen for sodium. That's essentially what I just said there. So that's what the antiport does. It just exchanges sodium and hydrogen. And then when we have the sodium bicarbonate, from the glomerular filtrate, decomposes it into sodium and bicarbonate. So it's essentially separating the bicarbonate from the sodium, where the sodium can then be pumped into the tubule cell. So when we've got an excess of sodium in the body, and then the ADH is telling the body that it needs to essentially get rid of all the excess salt that's in the body, it'll do this by separating sodium and bicarbonate and then excreting the sodium out with the urine. So bicarbonate then reacts with hydrogen from tubule cell to form this H2CO3 byproduct. And then carbonic anhydrase on the brush border, so this here, decomposes H2CO3 to water and carbon dioxide again. So essentially there's a whole lot of processes taking place so that we can then reabsorb water back into the body system or into the blood capillaries when we start to sense that blood volume might start to drop, and then therefore the blood pressure is also dropping. So carbon dioxide then enters the tubular cell and H2O passes in the urine. So this carries hydrogen that was originally in the blood. I'll leave you in your own time to go through this slide here. It'll make more sense for you if you read through it slowly and then going back through what all of these other buffering systems are that are taking place to try and get hydrogen from inside the blood to outside the body with urine. Alright, that was it. Thank you guys very much. I'll see you tomorrow afternoon.